Next lecture of the day, we are now going to one other aspects where cellular membranes are very important, and that concerns signaling. In fact, the membrane, in this case, it's a dilemma, right? If you want to receive signals from the outside, or you want to give information to the outside, you have a barrier. But on the other hand, if you make this membrane selective for receiving or giving signals, you have a type of filter. So only those information that maybe for this cell is relevant is then really able to cross the membrane and would, for instance, induce a cellular change, right? So let's have a look how this is realized in the context of organisms. So let's start with us as a type of multicellular organism, right? So our whole body is composed of over 200 different cell types, nerve cells, heart cells, liver cells, and some of them need to do cell division. Others need to grow, or cells need to differentiate. Some cells need to die. So in such a complex organism like us, where we have this enormous amount of different cells, it's only possible to survive when the cells start to communicate with each other. So without basically communication, multicellular organisms, it's not possible. In particular, specialization of a cell is not possible. So these cells that we have, they are very specialized. Those cells are important for signaling communication between them. Those cells are important to have basically the muscle construction of the heart. So each of the cells, very specialized. Liver cells are a type of chemistry fabric in our body. They are very specialized of breaking down material and making material. So definitely without cell signaling, no multicellular organism would be possible. No specialization would be possible. And the signaling allows the coordination of cell division, growth, differentiation. It allows the fight against pathogens to the outside. And it's important for sensoric and neural processes. Makes specialization possible. But most likely, this started earlier, already on the unicellular level. So already bacteria actually are able to talk with each other to signal. One example would be the biofilms. So maybe you have seen on lakes, depending also from the weather conditions, you might get actually a type of thin layer, which often are bacteria that start to aggregate. And that's sometimes for a particular period when in the year when they are basically well growing, and for particular conditions of nutrients, they start to form these biofilms. So already bacteria actually need to talk. So what we need to understand is how do they talk? What is the language of cells? How does this work? So first of all, basically, cells talk with the help of molecules that are basically released or accepted by them. And you see here just the diversity of molecules that can actually serve as signal. In our body, growth hormones or insulin, you know that insulin is, for instance, an important factor in our body for the regulation of the blood sugar level. Insulin would trigger the uptake of glucose from the blood into each of our cells. So obviously our cells recognize insulin as a signal to take up glucose. What would we require to take up glucose? Do you remember? What would be important then in the cell? Because we have a membrane, glucose, not very well permeable. So we would need a transporter, a glucose transporter. So this glucose transporter would be then basically taken to the plasma membrane in a response to insulin so that the cells are able to take up the glucose and can use it for your metabolism. You see basically that you also have type of lipids. That's a steroid. Lipids can be signaling molecules. No? Or you see like small chemical molecules here that are more derived from an amino acid, like adrenaline can serve. But even such small molecules like gases, even gas can be as a signaling molecule. That's astonishing, no? So what would be the structure that recognizes such a small gas molecule as a signal? That was puzzling over many, many years before people basically found a protein that is able to bind this gas and to actually use this as a signal. So you have quite a diversity, and now it depends, of course, from the type of cell, from the type of organism. What other signals can you think about? What about bananas? I had here two helpers that were so kind to actually buy some bananas for me. And if you let these bananas lying, what happens to these bananas? They turn yellow, no? Do you also maybe know from your parents that the parents would say, don't put the new bananas close to the old bananas? What is the reason?
They influence the other bananas to ripe faster.
The other bananas ripe no faster. What's going on? They talk with each other. The bananas that are old talk to the bananas that are new. And they do this via a gas, ethylene. And that gives this nice smell when you have in a house and you have these bananas that are very mature. You can actually smell this ethylene almost. So bananas talk. Or maybe you have heard about butterflies that can receive signals, pheromones, over kilometers. Can you imagine? Very few molecules, chemical molecules, that are recognized by a type of structure in these butterflies as a signal. Amazing. Very low concentrations, very, very sensitive sensors that can do this. So nature uses a lot of different type of molecules to serve as signals, and it depends, of course. We cannot detect these pheromones because we don't have the machinery to detect them, but in the butterflies, and some of the butterflies, they can recognize, and they can, of course, only recognize from their own species, these type. No? Okay, so we call basically these signaling molecules that are exchanged between cells so-called first messengers. And you will see the reason why we call them first messenger later. So in all cases, basically, we have here the cell surrounded by the plasma membrane, and typically it's a membrane protein that can bind and recognize this first messenger. And when it does so, it will change the conformation. So maybe like click. So it will change the conformation. And that typically leads even the change of within the whole protein that gives then a possibility to the cell to recognize something has bound to the outside because the inside has changed. And now basically this change triggers an intracellular cascade. So now comes other molecules that are activated and transmit the signal, and that can basically then end up to molecules that, for instance, change gene expression. So for those that are more in biology, it would be proteins that can bind to the DNA and either activate the gene expression or not. We call them transcription factors. Or it could be an enzyme that is involved in metabolism to produce, for instance, a particular new molecule that is required. Or it could be a protein of the cytoskeleton that will change now the arrangement and it will change the shape of the cell. So that we call basically the effector proteins that would be then change their activity depending whether a signal has bound to the outside or not. And what you also see is basically that since you have this type of cascade, you have here the possibility of an amplification. We will come to this a little bit later. Okay, so now, very simple. You of course, are welcome to dive even more than if you are fascinating about signaling into this topic. Let's have a look on these proteins that serve as receptors. And you can group them very, very roughly. It's very rough. You can group them in proteins that look like that. We call them G protein coupled receptors. This would be proteins that actually interact with another protein, which is a G protein. It could be these type of receptors that carry an enzyme activity. So those receptors by themselves are enzymes that can be switched on and switched off. That would be a receptor tyrosine kinase. Or it can be basically proteins that help to recruit other proteins, adapter proteins, okay? It can be an ion channel. We talked about this yesterday. An ion channel can bind a ligand, open or close, depending on what type of ion channel we are talking about. And a family of receptors we are not going to talk about today would be, for instance, receptors that are intracellular. But those would normally only recognize a signal when this signal can cross the membrane. So what do you think? What molecules would be able to cross the membrane? Small molecules, no? And if you go back here, sorry, from this basically here type, no? This would be a lipid-like molecule, right? And if it's lipid-like and maybe uncharged, it can also do flip-flop, and it could go in. So steroids are relatively easy to flip-flop, steroids. They have this very small head group, very large hydrophobic backbone, this steroid. And that's why steroid hormones, they are basically able to cross the membrane without help. And that's why the receptor can be inside. So that's the reason why for some cases you can have basically the receptor inside. But what we want to discuss is this family of receptors here on the plasma membrane. And we will only focus on 2 examples, just that you get an idea of how do they work. Okay, so one other important aspect is we have not talked about the molecules on the plasma membrane, the receptors. They can be grouped in different families. The other thing is that inside of the cell you have molecules that are, we call them molecular switches, almost like the light. They can be active or inactive. And there's 2 families of those proteins that work as switches. There are proteins that can be phosphorylated and dephosphorylated. And we have already talked about phosphorylation. It's always a drastic event because you transfer a charged molecule, phosphoric acid. And this type, then the protein becomes the area where it has phosphorylated, 2-fold negative. And you can remove it. And the protein that control these phosphorylation, dephosphorylation are kinases and phosphatases. So you have here a switch that is off when it's not phosphorylated, on when it's phosphorylated. And you can control this by a kinase that is phosphorylating and a phosphatate that is dephosphorylated. So you can switch between an on and off state. The other family, it's the so-called GTP binding proteins. So these are proteins that can bind the molecule and can change this molecule, and that switch between on and off. Let's go a little bit more in detail. So let's start with these G proteins. So these G proteins, they can bind a molecule that is called GDP, and they can bind GTP. So typically they bind GTP, and they have the ability to change this GTP into GDP. When they bind GTP, which is basically analogon to ATP, then they are active. When they basically hydrolyze this GTP to GDP, then they are inactive. So again, they can switch between an on state and, how do we say, GDP, phosphate, on state, off state. Then, of course, this molecule needs to leave, the GDP needs to leave. Now comes new GDP in, on, off. Okay, that it can do. It needs eventually to change from one condition to the other, also help us. It needs basically helper proteins to get into the on state, to exchange the GDP against the GTP. And it needs basically a helper protein to be activated to hydrolyze the GDP to GDP. Okay? And these helper proteins, these are basically here just given the GTP exchange factor and a GTPase activating protein. You don't need to remember this now for this course. It's only for those that are very interested in. Important is that we have a switch that can change between two conditions, and this switch is controlled again by helper proteins. There's even another helper protein that can move these G proteins in an inactive state. Okay, so what we are doing is basically we have at the plasma membrane receptors that can bind the signal, they can change the conformation, and that triggers here an event in the cytosol. It activates switch proteins. It can basically activate switch proteins that are phosphorylated, dephosphorylated, or it can activate switch proteins that are binding GTP and then hydrolyze the GTP to GDP to get inactive. Okay. So here's this example for the phosphorylation. Protein that is in the off state gets phosphorylated in the on state, a molecular switch that can then be switched back to the off state by removing the phosphate on the protein. Okay, now comes one thing more. These phosphorylation steps can also be coupled. Before I basically go through this, it's maybe easier to play. The point could be the following. I would be basically now a protein that gets phosphorylated. But I by myself have the ability to phosphorylate now another protein, meaning I would be a kinase. And Tanmay already sees that I look in his eyes to come here. Okay, so Tanmay could be just another protein, but he is my target. If I get phosphorylated, I could be a kinase now, that gets activated by this phosphorylation, and Tanmay I would bind ATP and transfer the ATP to Tanmay. Then he could be active. But what is if he would be now another kinase that gets active? Then you would come into play because you are the target of Tanmay, and Tanmay would activate you. So now we need even another box. Tanmay activates you and transfers the phosphate to you. And what is when you are also now a kinase? But in the moment we only have done 1 to 1 to 1. Now Tanmay, we have several copies of Tanmay. Do you want to be Tanmay second? So you have to imagine this is now Tanmay again. Stay here. Okay. So I'm a protein that gets activated by phosphorylation. I have the ability now to phosphorylate other proteins. These are my two targets there, Tanmay, Tanmay. So I would basically phosphorylate you. I would phosphorylate you. Now only one protein has activated two proteins. Now you both are activated and would have 10 times, what was your name? Hindon. 10 times. So then you could start to activate, you could start to activate. You see, so you get an amplification because what is drawn there, it's a little bit misleading because there's always several copies of the same protein. Maybe Thomas is existing 3 times. Tanmay is existing 10 times. Hydron is existing 100 times. And all 100 molecules could be phosphorylated only by 1 or by 2 or by 3 of those guys. You see? So it is as if basically your target would be all the others. And you would activate now all the others. Okay, and we call this a phosphorylation cascade. A phosphorylation cascade that allows to activate not only one by one, but that one molecule can activate several thousands of the substrate that are present. And so on. Okay, thanks a lot for that. Put this here.
In an exponential pattern.
The question was, Thomas, can this be in an exponential pattern? Huh? In principle, yes, because it's determined by the number. Now we have here a number of people that can actually do nice modeling. No, you could do now a modeling. You could say, okay, I have 10 copies of this, basically, of this molecule. Let's have 10 of those. No, how would be the signaling then? Or do I have 10, 100, and I take here 1,000 of those molecules? How would be? And if you have enough, basically, then it would be exponential. Then it would, no? It's not always realized like that, but in principle, yes, that would be a very fast amplification of the signal in an exponential way. Okay. So now, yes, please.
Where would the energy for the amplification come from? Obviously, you're increasing the energy, right?
Yes,
What
yes.
is consumed?
Very good point. The energy comes from the ATP, right? So it's stored in the ATP, and whenever you basically now use the ATP to transfer the gamma phosphate, you hydrolyze the bonding. That was what Tripta explained yesterday. And this gives a lot of energy that allows a new bonding to be formed. So it's from the ATP. Yeah, that's important. Okay, so that would be this cascade. So you would have eventually only one receptor activated. But then you can, in the end, that results to an activation of hundreds or thousands of copies of the same protein inside. Then another important aspect is that cells inside also use signals, so-called intracellular signals, second messengers. And those can be of very different flavor. It can be membrane-anchored second messengers that are produced after a signaling process, or it could be soluble second messengers. So calcium, for example, we have learned it's a lot of calcium outside because our cells outside have basically the type of ocean water composition around. But intracellular, in the cytosol, the calcium level are kept very, very low. If you now open a calcium channel and the cytosol is flooded with calcium, the cell definitely sees a change. In particular, to those proteins that would be able to bind calcium. If you have no calcium, the protein would be in one conformation. If no calcium comes in and it can bind it, a change in the conformation of this protein would change the activating status. So that's basically molecular switches that are used based off phosphorylation, dephosphorylation, based off binding of GTP, G proteins, and intracellular second messengers are used for the processing of the signals in the cell. Okay, now we have basically one point. We have now talked about the molecules, like the receptor and intracellular molecules. Now, of course, signaling occurs eventually on many receptors because the cells receive many signals at the same time. If basically two signals are received by a cell that are recognized by the two receptors that are present in this cell, it will lead to an event, a cellular signaling event inside of the cell. But it can cause a problem. We can also play a little game. I will say a sentence to you, and you transmit this sentence to the next people, okay? Yesterday was a cold day with bad weather. So I gave a signal. We could make it in the cell, it has to be fast, so we can make it, this game now very fast and stressful. Now you need to be faster, faster, faster. Yes, fast, fast. And we have some noise talking also eventually from the outside. It gets now more difficult because there comes a signal from another side. There could be a signal here. I need to go home. And this goes now there. And where's the other signal going here still? It's not going anymore. What's wrong here? Does anyone, did you receive any signal there?
Yeah, but not
What was it?
quite clearly.
Try the best. What did you understand? It was a cold day with bad weather, was the sentence, and this ended up. oh, what went wrong here in this cellular communication? That should not happen in the cell, right? But can you imagine now? And then you get another receptor telling also another message. And we have not talked about really the cytosol is full of proteins, right? When you did these GUVs, they were just in a way empty. No? There was only buffer in. But in reality, even now you are not on the level of a cell yet. We would need to invite probably 100 more people here in this room. And then it gets really crowded. And then if I give a message, it will be very hard to give this message for Harschmidt, right? Because maybe you need to find Harschmidt, no? How do you do this when this is your 100 people in? Maybe you do this here on the color festival or whatever. Maybe you also want to find your friends. Now you have the mobile phone, of course, you try to find them, but sometimes it might be very hard. So so basically there can things go wrong. And the cell needs, of course, to avoid this. And there are some tricks cells can do this. So this is what I want to here illustrate. You can basically combine the signaling molecules that should be activated by a helper protein, by a scaffold. So you keep the partners together that should start to communicate, right? Also here, she was not sure what message she should actually receive. Yeah? Eventually there came the message also from that side. So let's say, for instance, in some cases, cells in our body need to decide whether they should live or die. If you remember, for instance, our fingers, how would this look like in a baby when it would be still unborn? At a certain moment, there would be here still cells. It would be actually a clump of cells. And those cells here, they decided to die. They say, I will die. But such a decision, of course, you should be very carefully decided. Because if you decide here to die, there's no way back. These cells are dead. So then it becomes very important that the cell makes the right decision. So like in your life, when you want to make a decision, do you sometimes need to make decisions? Normally, how do you do this? When you need to make a decision, you sit in your room and then, what should I do? How do you do this? Can be that, you just say, do you do it like that? Okay. Okay, I tell you what I do. When I was young, I had a good relationship and still have a good relationship to my family. I went to my father and I explained to him. Then I went to my mother. I didn't talk to them, father. Yeah. My mother said, no, I think you could do this and that. And the father, no, I think you. And then I thought, also by myself. So you basically get input from several persons. Can be also your best friend or whatever. And then you make a decision. And say, do the same. If you have a critical decision like life or death, you don't want that, someone tells you, you are on the place to be, no, you don't believe this at the first instance. You need another signal, which also says the same. And then you might say, okay, obviously I'm at a place where it's important now that I should be removed. And this is in the same. So you have here basically a signaling, intracellular signaling molecule that needs 2 events of phosphorylation before it is really activated. It's not enough just here to activate Tanmai with 1 phosphate. He would be not active. He would need another phosphorylation event, and now you would be active, and now you would continue to signal to other partner molecules. No? Okay, so another point is, of course, very briefly, a decision like life and death, it's basically only 2. There's nothing in between. There's not half life living. No? So how do you turn basically an event into something all or nothing? Because when you have receptors at the surface and signaling molecules bind, one receptor is activated, the other copy of a receptor is activated, and another, and you get more and more activation. But how do you move now this stepwise activation into an all or nothing decision? Those that are very familiar with mathematics, what would it be to go from something gradual to something drastic? It's a type of step function. Step function that you need. So how do you turn something into a step function? No? And one way would be, for instance, you have this type of positive feedback loops, a self-amplification of the signal. So already a small change then triggers basically extreme, like an exponential increase of the signal to get this extreme fast response. Yeah? So they are basically more complex interplays of the molecules to allow gradual changes, but also to decide to allow all or nothing decisions. Okay, good. So now let's take the time for 2 examples. Just it's more like to get a flavor. Let's talk here about this first receptor family. So you see it's a membrane protein. Remember it was a polytopic here, no? Polytopic membrane several times. And in the case of this G protein, it's exactly 7 transmembrane domains. 7 transmembrane domains that span the membrane. And this protein can interact here with another protein that we call a G protein because it has the ability to bind GTP. So here's the receptor. And you see it has an extracellular part, a membrane-embedded part, and an intracellular part. This part here, the extracellular part, it's typically used to bind the ligands. And this part is used to interact with the G protein. And you see that in reality, of course, you have a 3-dimensional structure. So these 7 membrane domains are basically arranged in a way to get a very compact molecule that has here a binding site for the signaling molecule that comes from the outside, and here the binding site for the interacting protein, the G protein. And depending on what subtype of receptor of this class you have, the ligand binding pocket might be located here, might be located there, might outside or here. And that basically is depending now, if you have a receptor that recognizes monoamines, then it's more receptor where the binding pocket is like that. If it's a peptide hormone, the binding pocket is more located like that. If it's a hormone receptor, often the binding pocket is located like that. And then you have, for instance, here even receptors for glutamate. So you have the same, always the same type of receptor, the same building principle, but they can have different ligands. One receptor of this class might have the binding pocket close to the membrane and would bind the monoamines. Another member of this would have the binding site actually on a loop, on an extracellular loop. So that's why these type of receptors can have very diverse ligands to bind. And it depends now from the cell which receptor would be present, whether they react to one particular hormone or not, or to one particular stimulus. Okay, so now we keep it simple. We say this is our receptor that can bind a ligand, and that leads to the interaction with this G protein. And you see that this protein, again, is composed of subunits. That becomes now complicated. Now we have basically a receptor. This receptor interacts with a protein that by itself consists of 3 subunits, alpha, beta, gamma. And then you see that the alpha unit, it's linked via a fatty acid, and the gamma unit is linked via a fatty acid to the membrane. So how would we call these type of proteins? We call them peripheral proteins, because they are only not integrated, but linked to the membrane, attached to the membrane by this anchor. Okay, now this unit here, the alpha subunit, it's able to bind GTP in the exchange of GDP. And it would do so when the receptor gets activated. So let's have a look here what happens. So the first thing is, basically we have our inactive receptor and we have the G protein. You 3 want to come to play again? Okay, I see you want to be the G proteins, the G protein here. You want to be the alpha subunit? So Tanmay is the alpha subunit. What special property does he have? He can bind GTP or not. In the moment you are having not GTP, but GDP. So you are beta gamma, and in principle you can clump together, you are together. Yeah, that's okay, fine. What else does Tanmay have? He has an anchor, a lipid anchor. So you would need to have one hand for the moment up. Don't worry, not the whole, but then we have the beta gamma subunit. What would be the property of the gamma subunit? Having a fatty acid finger up. So you are anchored. And you are in a close relationship. So yes, exactly. You will always carry him with you, exactly. So now, okay, this protein, so what would this protein, if we do this now for the model membrane, if we would purify, this is a complex, 3 proteins, they would stick together, as long as we have here our GDP bond. What would these proteins be able to do in a membrane? They would move. Can you move? But you need to move together, no? All 3. Why can they move? Yeah. And don't forget, it's a random move. You are not doing random move in the moment. Now it becomes better. Yes, you can imagine as if you would be not knowing where to walk. Okay, why they are mobile? They only have the fatty acid in a lipid bilayer, meaning the lipid bilayer is fluid. Like lipids, they can move like a lipid, but they do it on the surface of the membrane. They would move. Okay, so I would be now the receptor. What would I be able to do? Also move, right? Oh. Sometimes I could come to you, sometimes not. Sometimes I can come to you, right? And they would also move. Okay, so that's nothing spectacular. Until the moment I would bind a ligand. Let's bind no ligand. Now I will be a helper protein for those guys here. I would activate Tanmai. And that would lead to the fact that he would exchange the GDP that he had against GTP. Now he is active, and what happens is he changes his conformation and can leave. Exactly, and now you can walk, and you can walk. So the complex has dissociated into 2 subunits, alpha, beta, gamma. And they can be basically now separated. So they can do things. They can, for instance, go to those guys there. And you could go here to this side to interact with another person, right? And that would stay like that, but in this case, for these proteins, Tanmai has the activity to hydrolyze this GTP to GDP. Okay? It's now difficult to show it as an example, but he can basically eat up the phosphate, and he will do this slowly. So once in a while, one of these alpha subunits would basically, the enzymatic activity would eat up, and then now you have GDP. And that would change, yes, that changes conformation, which when these guys meet him again, makes him sticky to those guys again. So, no? So now they will be in the resting state again. But I still have my ligand bound. So what will happen now again?
The ligand will release.
No, could be, but I'm in the moment, yeah, could be. That would be then the next step. But as long as I have the ligand, what is then?
You can activate it.
I will activate you again. What will then happen?
Change my conformation and move away from it.
So as long as I have my ligand, I will always reactivate. So what happens is basically now you slowly, now after a while, now is a while gone, what happens? You destroy this, and that gives you GDP. What happens then?
Conformation change.
So the G protein resets itself. So it will be activated and goes into the inactivated, but then they will meet me again, and as long as I am as the receptor, I'm the so-called GEF, the GDP exchange factor, the helper protein, as long as I activate Tanmai again, the G protein will be again basically activated, fall apart. That is what's happening. And only, you are right, when the ligand will dissociate and you will meet me again, then I cannot reactivate them anymore. So that's the principle of basically a receptor that works with a G protein, a protein that is able to bind GTP and has the ability to hydrolyze, but it does it basically only after the while. We call it a slow hydrolysis. Of course, if you have only one molecule, the hydrolysis by itself, it's fast, but it will not happen instantly. It will happen after a delay. Okay? Okay, thanks for that in the moment. And this is a little bit what is now here illustrated on this drawing. So you can go at home once more. You have basically here the inactivated complex with the alpha, beta, and gamma bound with the GDP. Now the ligand binds this receptor, activates the exchange of GDP to GTP, and that triggers the separation of the two subunits of this complex of this G protein into two individual molecules. Okay? And that's basically here also according to this list. The GDP is replaced by GDP. The activated beta-gamma complex dissociates. And then basically the G-alpha, so the alpha subunit and the beta-gamma subunit, they can now interact with other proteins. And then the point is that the G protein can reset itself, but as long as the receptor is active, it will be reactivated. And that goes basically as a cycle. So now what target molecules? I give you just one example. A target molecule could be another membrane protein. In this case, it's an enzyme, an adenylyl cyclase. And when this alpha subunit binds to this enzyme gets active and turns basically ATP into cyclic ATP. It's actually from the drawing here something missing. It should be another bound. So it goes from ATP to cyclic AMP. And this cyclic AMP would be a new water-soluble molecule that would flood in the cell and would have a target protein, for instance, a transcription factor that then can activate gene expression. So from that you get a signal outside that says this triggers cell division. When this signal comes, could be a type of growth hormone. The cells get the signal, turn on gene expression, produce proteins. Okay. So here's the example. The cyclic AMP binds to, not directly to the transcription factor, but to a protein, which is a kinase. And here's one aspect that I want to discuss just shortly with you. You see that this kinase, again, is actually built up by subunits, two identical subunits, a catalytical subunit that would be able to phosphorylate proteins and a regulatory subunit. And this regulatory subunit would, as long as it's bound, would keep the catalytic subunit silent. And only when the regulatory subunit dissociates, you get the catalytic subunit active, and that would then phosphorylate the protein, transcription factor, that gives transcription. But there's one interesting thing. You see that basically the binding here, the activation of the protein, the activation of the regulatory subunits requires the binding of 4 cyclic AMP. What does that mean? Let's say you would have here regulatory subunit, 2 binding sites, regulatory subunit. The cells produce 1 cyclic AMP, nothing happens. Now the next cyclic AMP is produced, still nothing happens. Now the next cyclic AMP is produced, nothing happens. Only when the 4th cyclic AMP, then you get activation. So you need a threshold of concentration. For those that are thinking about mathematics, if you need 4 to get the activation, when you have this threshold, that's actually a prerequisite for also these nice step functions. So you need this cooperativity. It's not enough 1. If the cell by accident makes a little cyclic, 1 molecule of cyclic AMP, you need really 4. So the cell really needs to be flooded with cyclic AMP that all the protein kinases that are depending on 4 molecules of cyclic AMP to be activated are really activated. Okay? So it's a type of, if you want to say, sensor system. Whether there's enough concentration, this system would not be linear. It would not react linear with the concentration increase. No? Okay. And this example you find actually in signaling at many places. You need not only one copy of the signaling molecules, you need several to get the next step activated. Okay, and then this was basically, now we can here phosphorylate another protein, for instance, a protein that interacts with the DNA and tells basically the polymerase here to make the messenger RNA so that you start protein synthesis. Okay, so now we have basically just talked a little bit on these G protein coupled receptors. Let's take this class here, those receptors that are basically having an enzymatic activity, okay? Now, you see already that they look a bit different from the topology. Obviously, those have two single-spanning transmembrane domains, okay? And in fact, they might be in the membrane as two proteins. And only when they come together, they would basically get the enzymatic activity, meaning since they are kinases, they would be able to phosphorylate, and they could actually phosphorylate themselves, cross-phosphorylate. So why is this an important step? Or maybe before we come to this, just here for the chemists among us, so that would be here our, for instance, tyrosine kinase receptor, and then you see there's a part of the tyrosine kinase receptor that contains the amino acid tyrosine. And this amino acid can be basically phosphorylated, and in the tyrosine kinase receptors, this is a major event. So when this receptor gets activated, the receptor itself gets phosphorylated on a tyrosine, and that's why they carry the name. And you see when you do this, this would be polar uncharged, this would be polar supercharged. So you drastically change in the protein itself, again, a part that would be highly charged would, due to the interactions with the other part of the protein, lead to a conformational change. Okay. So you would basically have then the possibility by the phosphorylation of this receptor that you get a change in the environment, in the intracellular environment, that serves as a signal for other proteins to dock. So you create a docking site. And basically this autophosphorylation, if you want to say the transphosphorylation within the receptor, creates docking sites. And the receptor by itself, after having docked this protein, can also phosphorylate the target so that on top, the target eventually gets, while it has docked, activated. So you have two tasks of this phosphorylation event. One task is that the receptor gets activated serving as a docking. And the second task is that the receptor itself can activate by direct phosphorylation of the substrate and thereby activate the substrate. Okay, before we come to the two examples, there's one aspect that I want to discuss with you. Tanmay, you want to come? So Tanmay and me, we are the receptor. We carrying here the activity to be able to phosphorylate other proteins, the kinase activity. And normally we would be basically just as monomers. Okay. So now you have to imagine this is the part that is outside of the membrane, this is the part that is inside in the membrane. And I would be only a single spending membrane protein. Let me see. Maybe I can use this. No, this here. So what would I do as a single spending membrane protein? What can I do in the membrane? If you think about the membrane again, now let's go back to the fluidity of a membrane and so on. What can this membrane protein do? I can do this, of course. I can do this. I can do this. All these movements you can do. And I can, of course, in case of a receptor, I can bind here something. What do you think would this mean for the part that is now intracellular? So now I have not bound anything on that part, and I move, and I bind here something, and I move. What would this mean for the inside part of this protein? You think so? It's looks like I look like that. here has something changed. But if you are so flexible, no? Remember, this would be more like, you can hardly do it. If you are so flexible, there will be hardly any change. We can discuss, of course, do I change my diffusion speed now because I'm heavier? But how much change is it? And so it will be very hard to see for the cell inside that something has changed. But what if this binding would trigger something like that? Does then has something changed inside? A complex has formed. That can be seen from the inside, right? I'm alone. Tanma is alone. Now I bind and you bind ligand. Now we come. That can be seen. And now on top we have an enzymatic activity. I phosphorylate him, he phosphorylates me. And that attracts now adapter proteins. So, thanks a lot. A single spending membrane protein cannot signal alone. It needs to form a dimer. Okay? So these receptors, this is in contrast to these G protein coupled receptors. Remember, they had 7 transmembrane domains. They were quite complex. So there were 7 transmembrane domains. If you bind something, that will change their arrangement between. But with a single membrane spanning protein, there's no, if I bind something or not, there's hardly any change. Diffusion rate, maybe a bit heavier, but typically the ligands are also very small. So then you need this drastic event of dimerization. So these tyrosine kinase receptors, they need to dimerize. That's the only way how they can signal because they are single spending, no? Biotopic membrane proteins. Okay, so, but there's now 2 things, 2 principles even within in this class. So we can talk about a so-called epidermal growth factor. That's a receptor that tells the cell, you should divide. And we can talk about the insulin receptor. That's the receptor that tells the cell, send glucose transporters to the plasma membrane, take up glucose. Huh? Okay. So in the case of the EGFR receptor, we have this example that we just were dancing. We have basically 2 single spending transmembrane copies that need to dimerize. And the dimerization is triggered by the binding of the ligand. And this basically then triggers the activity of the enzymatic part, the intracellular part, for the phosphorylation. And by that way, you get the receptor activated. Now the receptor is activated. Okay? And in former times, let me see. Yeah, in former times, you see this still in old books, because we had not the structure of this receptor. There was one idea about how they come together. Tanma, I'm sorry, you want to play again? Okay, so, so what do you think? If you now add to S a ligand, what could be the reason that we come together? I mean, anyway, once in a while we come together, right? Because we diffuse. Once in a while, but this will not keep us together. What do you can come up with ideas? I just want to trigger you, that because that helps you a little bit when you think about problems. So the problem is to understand how can the binding of a ligand keep us then together? Do you want to come up? You can take a moment time. We play again. You also moving still. No? We are a dynamic membrane. The others can still think. Once in a while we meet, but there's no reason for us to stay together. No? So why would a ligand binding now trigger us to stay together? Okay, one thing that people came up, could it be, because we have 2 binding sites, no? Just look a little bit around here. Maybe, yeah, Tanma here. Could it not be like that? How do we call it? The ligand cross-links us. That is still what you see in books often. They draw it like that. This ligand binds on this side, no? And on that side. Could also be because the binding is identical, we are identical, no? And now we are together. That would be, and that could be a good idea. And you only know when you know the structure of the complex with the ligand. Only then you can tell. And when you have the structure, then you see that this model is wrong. That is not how it's working. Because I need to bind, you need to bind, but our binding sites are not the sites where we bind together. So when you bind and I bind, we change basically this part here, the extracellular part, a bit. And actually, I need your back. If you turn your back to me, yes, that is fine. And actually, Tanmay already says like that. He binds the ligand and he does this a little bit. And I have the ligand and I do this a bit, and then we come like that together. It's a complete different picture, right? Instead of actually binding like that. This would be cross-linking. The ligand cross-links us. But what the ligand does, it triggers a conformational change that leads to a stickness of us like that. And now we are still together. It's the same principle. We are together as a dimer and we can start signaling. But of course, this type of mode, you can only find out when you have the structure. But you can come up anyway with ideas, and then you can think about also how to test these ideas. No? Thanks a lot, Tanmay. Super. Okay. So that's why also there is a other class of this. They are already cross-linked. They are cross-linked. So we are already forming a complex. And if this is the case, now I don't take Tanmay again, he gets stressed. Can one of you colleagues come to me again? Yeah? Yes? So we would be always together. So like that, we would be always together. But once a ligand binds, we could change, like that. So a little bit like that. So and then here these domains come together. So, and then we can still fall. Because then if two parts are together, we can change relatively to each other the positions a bit. So we can, between us, there can be a change. And that can be recognized. So in this case, the problem is basically already partially solved by forming a dimer, a preformed dimer that only needs to change the conformation. In the other case, we need to form first the dimer before we get the signaling. Super. That was the important point that I wanted to make, that you get an understanding basically how the change of protein complex formation and disassembly can actually change a signaling in the cell. Good, not to go, this is really now, we talked about basically, then we have this phosphorylation that helps other proteins to get basically associated. And among those proteins can be, again, proteins that activate another subclass of G proteins. So that would be a G protein that also is able to bind GTP, and that would activate now kinases, a type of phosphorylation cascade. So one protein phosphorylated